I will begin by giving a brief overview of the clinical trial landscape. We will then have the opportunity to hear from some of the different clinical trials that are ongoing for FTD. But what exactly is a clinical trial? So, clinical trials are a research study that is designed to determine the safety and effectiveness of new medical interventions. These medical interventions can be for a drug, a device, or a new surgical procedure. Every clinical trial is different in what it asks of the participants in the potential benefits and risks that participation may bring. Before researchers can begin to recruit participants, they are required to define a protocol that is then reviewed by an ethical oversight board. This is because there are many laws and regulations that oversee and protect the privacy and confidentiality of those who participate in the clinical trial. Very briefly, there are several different types of clinical trials. There can be non-drug therapies, and this may be a lifestyle or behavioral change that might improve disease symptoms. There are drug therapies, which we often hear about, and this could be based upon a molecule or a chemical compound that has been designed to reverse or block disease-causing biology. There's also immunotherapies, and these are based upon the use of antibodies that might halt the disease. And finally, you may hear about gene therapies, and this is an approach designed to replace or modify disease-causing genes. So there are several steps needed for the drug development process in order for it to achieve safe and effective treatments. The journey from research to available treatments can be long and complex. It will start where there's the disease understanding stages, and at this stage, researchers are aiming to better understand the biology that causes the disease. Then there are the preclinical stages where potential therapies are being tested in test tubes, cell culture, and animal models of the disease. And if a therapy looks promising from these preclinical studies, it will then be tested in clinical trials, and a clinical trial includes several different phases where safety, effectiveness, and mechanism are evaluated. With clinical trial data collected, it will then undergo regulatory review by agencies such as the Food and Drug Administration. And that medical intervention will then continue to be monitored for its safety in humans in the long term. And you will have opportunity today to hear from several company members who are at different stages in this drug development process. There's active research on every form of FTD occurring along the drug development pipeline. This includes research on sporadic and genetic forms of FTD. Today, several companies may speak about their focus on the progranulin mutation. That is in part because there was an early understanding of the connection between progranulin and FTD, and the development of tools were made available to test this potential treatment. But as more genetic causes and we learn more about sporadic FTD, like progranulin, researchers will learn about the biology and the genes and become more confident in how we can tackle the underlying mechanisms. You may be wondering whether clinical trials from one type of FTD can inform clinical trials for other types of FTD, and the answer is yes. Clinical trials from one form or type of FTD will drive the progress for drug development in all forms of FTD. The experience and learnings from these more mature studies will have ramifications for all people impacted by the disease. We can learn about the disease biology that bridges all types. We can learn about biological tools like biomarkers that allow us to monitor the disease. And we can learn about what is meaningful to participants in FTD trials and how this could inform trialists on how to best recruit and retain participants, and much, much more. These learnings will fuel the future of FTD drug development for everyone impacted by the disease. And our community isn't learning just from FTD trials. We're learning even from diseases that are related or rare. For example, there's clinical trials that are happening in diseases that have a shared biology with FTD. An example of this would be clinical trials focused on C9orf72, the genetic mutation that can cause ALS. But it can also cause FTD. And so researchers are able to better understand this shared biology, and this provides insights on future approaches for drug development. We also know that it is possible to have and complete a clinical trial for rare diseases, and this was observed by the Wave Life Sciences company, who completed a C9orf72 ALS FTD trial. Further, other rare related neurodegenerative diseases have had successful approvals, and this was seen by Friedreich's ataxias. So drug approvals have happened in related diseases and can happen for FTD as well. And there's several ways that you can get involved in FTD research and clinical trials. You'll have the opportunity to hear more about this at tomorrow's FTD one-on-one session. It is my pleasure now to introduce you to each of the companies here ready to share what's going on in their clinical trials. First up, we have Dr. Richard Tsai, Executive Medical Director at Denali Therapeutics.
Hi, everyone. First of all, I just want to take a moment to thank the AFTD for giving the industry partners a chance to share our work and our thoughts, especially around drug treatment trials. So as Amanda said, I'm Richard. I am a neurologist by training, and I've actually been working on FTD first as a practicing physician for over 10 years now. But today, I will be presenting some of our work with Denali Therapeutics, especially on a clinical trial for FTD associated with the granulin or GRN gene mutation. I'm an employee at Denali, which is a biotech company. And just a note that this program is also partnered with Takeda Pharmaceuticals. But Denali is a relatively new biotechnology company. We are very much focused on neurodegeneration. So you can see on the bottom left there, we're focused on lysosomal storage disorders that have a cognitive or behavioral manifestation, Parkinson's, Alzheimer's, but most importantly, we also work on ALS and FTD. I guess the takeaway message here is that we are really focused on neurodegeneration. We have multiple programs in neurodegeneration, and that's what we do all the way from our scientists to our lawyers. We think about neurodegeneration all day, and we're here to stay. So specifically for FTD, Denali and Takeda, we are collaborating on a way to develop a potential treatment for FTD caused by the granulin gene mutation. So as described earlier, about 10% of all FTD cases are caused by mutations in one of the two copies of the granulin gene. And because of this mutation, it is known that a person affected is now producing less progranulin protein than normal. And when we measure it in the CSF or blood, we can see that patients affected oftentimes have 50% or 30% of the normal amounts of progranulin protein. And it is thought that it is this loss of progranulin protein that's causing all the symptoms in FTD. So our treatment hypothesis is actually really simple. If patients are missing this protein, why not give it back? But the challenge is proteins are a big, big molecule, and they're normally blocked by this thing called the blood-brain barrier, which is a barrier around the blood vessels in the brain that's normally there to protect you from toxins and things from going from the blood into the brain. But it also now prevents drugs, big drugs like antibodies and proteins from going from the blood into the brain. But Denali has developed this proprietary technology called the transport vehicle, and it's actually really simple. This blood-brain barrier normally also has a receptor that transports iron from the blood into the brain for the brain to use, and we're simply piggybacking on this mechanism to go from the blood into the brain. So we're utilizing a naturally occurring pathway to piggyback a drug as big as a protein from the blood into the brain. And the nice thing about this is that we can use a simple IV infusion, so say in the arm, to deliver a progranulin protein from the blood into the brain where we think it's most effective. And so far, we are active in a clinical trial that I'll describe in just a second, but the preclinical evidence, as Amanda said, usually before you go into trial, you study the drug in animals and cell cultures and things like that. Animal models that reflect FTD caused by the granulin gene mutation, we have been able to correct a lot of the abnormalities in these animal models that are also seen in patients. So this is the ongoing clinical trial. The drug is called TAC594 or DNL593, and this is an ongoing phase 1/2 clinical study. We're currently in the Part B portion of this clinical trial, in which we're recruiting patients who have the granulin gene mutation and have active symptoms. You can have any types of symptoms. There's no restrictions on that, and there's also no restrictions on the disease severity. As long as you have symptoms and the granulin gene mutation, we will allow. The treatment period is Part B. It's the double blind, so that means there's a placebo and the active treatment. The more patients will get the active treatment than placebo. This double blind period is for 6 months. And after 6 months, there's an 18-month open label extension. And what open label extension means is that everybody will get the drug. And this study is currently ongoing. So just a quick summary. So TAC594 or DNL593 is an investigational drug. It's a progranulin protein that's designed to cross from the blood into the brain via an IV infusion. It's for patients with FTD caused by the granulin gene mutation. We have active sites in Europe, South America, and the US. That is the clinicaltrials.gov number that you can look up online to see if there's a clinical site close to you. We're happy to answer at our booth right outside this conference room. And again, thank you to everyone for attending today and AFTD for giving us the chance.
Thanks, Richard. Up next, we have Dr. Will Chu, who is the CEO or Chief Executive Officer at Passage Bio.
Great. Thanks to AFTD for having us here today. Will Chu, I'm a geriatrician and also the CEO of Passage Bio. And I'm going to talk to you today about PBFT02, which is our gene replacement therapy for FTD with both the GRN or the C9orf72 mutations. This is an investigational product, so any data I show you is early. It's very preliminary, and this product has not been approved for use by the FDA. So Richard already covered that progranulin deficiency is the driving cause of neurodegeneration in patients with FTD-GRN. So obviously, all of us up here are trying to replace progranulin. So our product candidate is called PBFT02. It's a one-time therapy, so you come once for an injection. It's a gene replacement therapy. So essentially, patients have a defective copy of this gene. We give them a functioning copy of it so that their own brain cells will create progranulin. It is delivered via a non-surgical injection directly to the cerebrospinal fluid. And I'm going to share data from our first treated patients, which has showed a high, long-lasting effect. So pretty high levels of CSF progranulin that have lasted out to 1.5 years so far. So first, a little bit about the route of administration of this gene therapy. So the delivery for PBFT02 is via the intracisterna magna route. So what is the cisterna magna? It's a pocket, a large pocket of cerebrospinal fluid at the base of the brain that is outside of the brain tissue. And some important anatomy here. So the cerebrospinal fluid, it bathes the entire brain and is in communication with every part of the brain. And so from anatomy, we believe that giving a product directly to the CSF should reach all parts of the brain. And in fact, we have good preclinical data that shows that our partners at the University of Pennsylvania delivered this product to non-human primates, and they were able to show that it reaches every part of the brain when delivered to the cisterna magna. The procedure itself only takes about 45 minutes. The actual needle in time is very short. 45 minutes start to finish. It's done in a CT scan and usually by an interventional radiologist. Okay, so this is a complicated slide. I'm going to make it real simple by talking about the key parts of our ongoing phase 1/2 study. So first, this is a multinational study. We have sites open in the US, Canada, Brazil, and Portugal, and soon we'll be opening up sites in Spain and Australia. Our US sites right now are Houston and the University of Pennsylvania. Very soon we'll be opening at Vanderbilt, and shortly after that at the University of Michigan. We are in the middle of our study of FTD-GRN patients. So we have scheduled to treat 15 FTD-GRN patients, and I'm going to be sharing data with you on the first 7 of those patients. Those first 7 patients received dose 1. So this was the original dose. We had always intended to go up in dose. That's what you do in phase 1 studies. You try and keep increasing the dose to find an effective dose. As it is, the dose has been very effective so far, so we're actually moving down to a lower dose. Dose 2 is going to be half of dose 1. So subsequent patients will receive dose 2, which is half of dose 1. The key biomarkers that I'm going to be talking about today are progranulin, so is the product doing what it's supposed to do, and an early look at neurodegeneration, which we are measuring by neurofilament levels, and I'll talk about that in a second. So our key eligibility criteria, it's patients aged 35 to 75 who have symptomatic FTD and are carriers of either the GRN gene or the C9orf72 gene, and you can see some of the key exclusion criteria listed here as well. Okay, so this is data from the first 7 patients who have received dose 1 of PBFT02, and this is the safety data. So in 5 of 7 of the patients, all adverse events were mild to moderate in severity. In 2 of 7 patients, they experienced serious adverse events, which required a hospital visit. Those included liver lab abnormalities and vein thrombus. All of these serious adverse events were asymptomatic and responded to treatment. There was no evidence of nerve toxicity and no complications during the product administration. And as I mentioned before, the remaining patients we're treating in this cohort are going to receive dose 2, which is 50% of dose 1. One of the reasons we feel very happy with moving to a 50% lower dose with dose 2 is the robust response we've seen so far from dose 1. So what you see here is progranulin levels in the CSF. This is what we're trying to do. We're trying to raise progranulin levels. So all of the treated patients started at very low levels of around 2 to 3. The normal level is around 3 to 8. From our preclinical work, we're targeting getting average values at least above the normal level. And what we've seen so far is levels anywhere from about 15 to 30 of CSF progranulin levels. So these are very high levels, and importantly, they've been durable. So they've remained consistent out as long as we've followed up patients. So our longest patient has been followed up for 18 months, and they show no sign of any waning effect. That's important for us because if this product is going to be a one-time genetic therapy, we don't want it to lose its effect. You want to come in just once and not have to come back again. The other interesting piece of data that we're starting to interrogate is on the biomarker of neurofilaments. So neurofilaments are a sign of degenerating neurons. And in FTD, neurofilament levels in the plasma are really the only biomarker where we understand what the disease progression looks like in untreated patients. So we have good natural history data for plasma levels of neurofilaments. And from this natural history data, untreated FTD patients who have the GRN mutation and are symptomatic have an annual rate of change of about 29% of plasma neurofilaments. And while it's very early in our study, the first 2 patients who've been followed out to a year have an average plasma neurofilament level of negative 13%. So far it looks like in at least these 2 patients, there seems to be an effect on neurodegeneration. So in conclusion, dose 1 of PBFT02 demonstrated a robust, consistent elevation of CSF progranulin. The progranulin levels remain elevated in all patients followed to date. There's early evidence of improvement of disease progression as measured by plasma neurofilaments when compared to the course of untreated patients. 2 of 7 dose 1 participants had serious adverse events, all of which were asymptomatic. And we are currently enrolling both FTD-GRN and FTD-C9 participants to receive dose 2. Thank you very much for your attention, and look forward to any questions.
Up next, we have Dr. Yiga Wang, the Associate Director of Clinical Development from AviadoBio.
Thanks, Amanda. My name is Yiga Huang. I'm Associate Director of Clinical Development at AviadoBio. My background is I'm a physician neuroscientist trained at Oxford University in the UK, hence my accent. So I'm going to be giving an update on Aspire FTD, our phase 1/2 study of AVB101, which is our gene therapy for FTD-GRN. So at AviadoBio, our mission is to develop potentially transformative gene therapies for people living with neurodegenerative disorders like FTD. And we believe what sets us apart is our combination of next-generation gene therapy design, deep neuroscience expertise, and neuroanatomy-led approaches to drug delivery. Now, similar to Passage Bio's approach, we are going past the blood-brain barrier. What differentiates us is that we're going directly into the brain. So I think the best way to explain this is through a video, and this is going to show how we restore progranulin in the brain.
AviadoBio uses a one-time surgical procedure to deliver gene therapy directly to the region of the brain called the thalamus. Cannulae with very small tip diameters are directed into the thalamus of the brain, and infusions are monitored using real-time MRI via co-administration with a contrast agent. The gene therapy is delivered as bilateral pairs of infusions to the target structures. The procedure is completed with the goal of achieving broad distribution of the treatment via thalamic connectivity throughout the brain. Direct intraparenchymal delivery bypasses the blood-brain barrier and peel membrane to allow region-specific targeted distribution of gene therapy.
Okay, so Aspire FTD is well underway, and our key eligibility criteria include having an FTD diagnosis and disease-causing GRN mutation, being of age 30 to 75 years, having a CDR plus NAC FTLD global score of 0.5, 1, or 2, as well as being able to undergo the administration procedure. Our key endpoints cover various aspects of safety, efficacy, and biomarkers. We have dosed our first cohort, and hot off the press, today we announced that we've finished dosing our second cohort as well. And recruitment is currently ongoing in the US, UK, and EU. So here's a map of our sites. Notice on the left-hand side in the US, the red dot represents dosing sites. So this is Ohio State University in Columbus. That's also a neurology site. And then the other two dark blue dots are our current neurology sites, so Vanderbilt and Houston Methodist. All of these sites are arranged in a double hub and spoke fashion, which means that we can leverage both FTD and neurosurgical expertise. So Aspire FTD follows a gene therapy trial design, and that is typical of gene therapy trials. And after screening and baseline visits, participants are given a one-time treatment of AVB-101, and they're closely followed up for 6 months, and then there's a 4.5-year longer-term follow-up. So Aspire FTD represents the first intrathalamic gene therapy delivery for any adult and for any neurodegenerative disease. And administering under MRI guidance allows us to visualize infusion and be really precise with our delivery. So I'm pleased to say that preliminary data from our first cohort shows that it's generally well tolerated, with adverse events being mild to moderate. In this cohort, there's no serious adverse events or serious reactions to the product. And no immunosuppression has been used for any participant. So in summary, AVB-101 is a one-time gene therapy aiming to restore progranulin levels in FTD-GRN patients. Preliminary data show it's generally well tolerated, and we're currently recruiting out sites across the US, UK, and EU. And I'm also pleased to say that recently we've announced a collaboration with Astellas Pharma to potentially accelerate development and widen global access to AVB-101. I'd like to say a big thank you to the FTD community, all of you, and a special thank you to our participants and caregivers for their time and commitment to our trial. Thank you.
Thank you. Up next, you will be hearing from Dr. Stella McCoy, Vice President of Medical Affairs at Alector.
Hi, everyone. My name is Stella McCoy, and I do work in medical affairs at Alector. To tell you a little bit about Alector, we're a company that are developing drugs in neurodegenerative diseases. And our phase 3 trial is in frontotemporal dementia, which is why we're all here today. I know a lot of this is background, but just to start with, we all know that FTD is the most common cause of dementia under 60, and it causes a really rapid progression in memory impairment, cognitive function, and behavior. And we're all here today, as you know all too well, because there's an extreme unmet need for individuals that are impacted with FTD. There are no therapies, approved treatments to cure or slow the disease, and that's why we're all doing the work that we're doing today. The session before talked a little bit about how FTD can be sporadic or genetic. Sporadic, just to reiterate, that means that there's not a clear inheritance pattern or a gene that's been identified that's causative of the disease. And in genetic forms, we do see inheritance patterns or causative genes that have been identified. The most common forms, as many of you I'm sure know, I see many people representing different groups here, are GRN, which our panel is talking about today, C9orf72, and MAPT, although there are many other identified causative genes as well. Okay. At Alector, we're in phase 3 clinical trials, and our phase 3 clinical trial is called INFRONT-3. This trial has completed enrollment, so it is no longer recruiting, but it is an ongoing global pivotal phase 3 study that's investigating our investigational drug called latozinemab in participants with FTD-GRN. You might ask what latozinemab is. This is an antibody therapy that's given via IV, and the proposed mechanism is that it blocks the degradation it blocks sortilin, which is a degradation receptor, in order to raise progranulin levels back to that of the normal levels. So it is a phase 3 study where we're following individuals for 96 weeks. And after that 96 weeks, there is an open label extension where the individuals who completed the first part of that study could roll into the open label extension, where everyone in the open label extension could receive the active drug, which is latozinemab. We enrolled 119 participants globally, and this includes both individuals that are symptomatic with FTD-GRN, as well as individuals who are at risk, meaning they carry a pathogenic GRN mutation, but are not yet presenting with symptoms of FTD. One other thing I wanted to highlight is you might ask, like, what does it mean that a study is pivotal? A pivotal study is used to describe a trial that's designed in such a way that in the case of positive data, the evidence would be supportive of a submission to the health authorities for consideration for approval, and that's what this trial is. We completed enrollment of this trial in Q4 of 2023, and that means so all of the participants have been enrolled, and we're just awaiting completion of this trial, and we expect data in Q4 of this year. This was an amazing effort that involved this community and a lot of people that are here. In order to complete this trial of 119 participants, we had 66 trial sites in 16 countries, and you can see that pictured here. It took 3 years, a little over 3 years, to achieve the recruitment of the study. And as part of that, we pre-screened 442 individuals, and 240 individuals were screened for inclusion and exclusion criteria. The 442 individuals that were pre-screened could have been things for, like, do they have the progranulin mutation? Do they have an elevated neurofilament level? And then the 240 were for other criteria that the individuals would have to meet in order to be a part of the trial. 103 of the 119 individuals in the study were symptomatic or are symptomatic, and 16 of the individuals are at-risk carriers. So we are anxiously awaiting the results of this trial later this year, but wanted to say a thank you to this community, to the families, to the Infront 3 investigators, and all of our sites who made this all possible. So, thanks.
Hello, everyone. I am Sophie Fransen. I am a research scientist, and I'm here today on behalf of Vesper Bio, which is a biotech company based in Copenhagen in Denmark. I am honored and very excited to be here and to tell you about our progress towards developing an oral therapy to treat frontotemporal dementia. So let me start by telling you why our work is so important. If I can click, there we go. So at Vesper Bio, our mission is to find new ways to treat neurodegenerative diseases. Our key focus is frontotemporal dementia, or FTD, which we all know is a disease with devastating progressive symptoms that worsens quality of life. It often strikes people in midlife, and today, sadly, there are no approved treatments on the market that can either slow or stop this disease. So there is an urgent need for new treatments. There are different forms or subtypes of FTD, and at Vesper, we are particularly focused on a form called FTD-GRN. And as we know, this is where people have a specific genetic variant that leads to much lower levels of a protein called progranulin. So our focus or our goal is to restore progranulin levels and also to help improve the lives of people living or affected by FTD. Progranulin is very important for keeping the brain cells healthy and also for protecting the brain against damage. But what actually happens when we have too low levels of progranulin? If we zoom in a little on this disease, when we have too low levels of progranulin, two main things happens in the brain. First, inflammation is increased like a fire that damages brain cells. And second, there is a increased cell death. There's an increased cell death that means that the function of the brain worsens with time. So these are the key features of the disease and are closely linked to the symptoms of FTD. So how do we target these problems with our treatment? Our treatment relies on the interaction between two proteins. One is sortilin and the other is progranulin. These two proteins can bind to each other and they play a relevant role in neurodegeneration. When progranulin binds to sortilin, this binding leads to lower levels of progranulin available in the brain. So our drug, which is called VES001, is a small molecule and is able to enter the brain on its own. Inside the brain, VES001 can block sortilin, so the amount of progranulin increases, which helps reduce inflammation and protects the brain. Our connection to sortilin research goes back a long way, as one of Vesper's co-founders discovered sortilin back in the mid-90s, so we have built more than 30 years of experience working with this protein. VES001 can be taken as a tablet in your own home, monitored by a caregiver if needed. So we are extremely excited about offering an oral therapy, as we hope that this will make the treatment more convenient, easier, and much nicer for the patient. So how far along are we with this new treatment? At this point, I am very happy to share that we have dosed our oral therapy, VES001, in healthy individuals in a clinical study that was successfully completed last year. In this clinical trial, VES001 was shown to be safe, well tolerated, and was able to increase progranulin levels in the brain. So this was a very important step forward, and we have now moved on to the next phase, a phase 2A trial, which is currently ongoing, and we are expecting results later this year. So at Vesper, we are very grateful to be part of the effort to make the lives for people affected by FTD better, and we look forward to sharing more information with you in the near future. Thank you. If you want to know more about Vesper or our work, you can scan the QR code. That was, yes, here. Thank you very much.
Thank you. Up next, we have Dr. Hamza Megdadi, Medical Director from Prevail Therapeutics.
Hi everyone, my name is Hamza Megdadi. I'm a medical director at Prevail, which is part of Eli Lilly Genetic Medicine Institute. So my colleagues are already talked about granulin in a lot of detail, so I'm not going to repeat that. I'm just going to add a few things. First of all, the gene therapy mechanism of action. First of all, we have three types. The in vivo gene therapy is what our clinical trial and my colleagues, the two other clinical trials in gene therapy that focus on GRN, they are all some type of in vivo gene therapy, which is a replacement gene therapy through a vector. And the reason GRN is the main target for our trials is, as mentioned before, granulin gene is loss of function. It leads to decrease in the progranulin level. So a replacement is hypothetically should lead to stopping of the disease. Other genes, they might need some type of gene editing to remove the gene, which is something like CRISPR, which is still early in the development, and not a lot of clinical trials have that, but in vivo gene therapy, there's already marketed products with approved products for other diseases. Okay. This is the function of the granulin gene. We talked about this. Just our therapy, we also similar to other clinical trial, we provide a healthy or a functional copy of the granulin gene through a vector delivered through the ICM or intracisterna magna, similar to Passage Bio. And it's one-time treatment with follow-up up to 5 years. We already completed the first 3 cohorts, and our first cohort results already published in Nature Medicine last year. And I'm not going to go through the details of our trial design, and if you have any question, you're welcome to stop by our booth. And we also have a genetic testing initiative with Cyanogenetics. This is for people who already have the diagnosis of FTD or have symptoms that might be FTD symptoms, or even at-risk family members who want to know their genetic status. So this is at-home genetic testing with pre and post testing genetic counseling. Our colleagues from Sano also are at our booth, and you're welcome to stop by and talk to them as well. Here are our clinical trial sites. We have trials in Europe, we have sites in Europe, Australia, and 2 in the US. And I would like to thank everybody, and please stop by our booth for more information.